For our clinical Correlate project, we will be discussing the effects on high versus low carbohydrate diets for athletes. Our presenters today will be Mallory Hanson, Elizabeth Jantosh, Audrey Warren, and Kendall Heller. Our audience will be able to learn these things after our presentation. They'll be able to identify when glycogen stores are used during exercise, identify critical transporters, and their roles in carbohydrate absorption. They will be able to list effects of a low carbohydrate diet on blood pH and identify the impacts of high carbohydrate diet on physical performance and body composition. Carbohydrates have many important functions in the body. The primary role of carbohydrates is to supply energy in all cells in our body. Many different mechanisms in our body utilize carbohydrates to fuel their processes. Carbohydrates help the function of the central nervous system, red blood cells, and tissues because they are all dependent on glucose as a primary energy source. Digestive operations is another function of carbohydrates. Insoluble fiber helps make stool softer, promotes bowel health and regularity, and supports insulin sensitivity. Soluble fiber improves digestion and reduces blood cholesterol and blood glucose. Digestion of carbohydrates begins in the mouth and is broken down into glucose. Then this makes its way through the stomach where digestion is halted, and it proceeds to be digested in the small intestine by breaking down disaccharides into monosaccharides. Absorption in the small intestine is active and carrier dependent. This then gets sent to the portal vein, to the liver, and then to the blood. The enzymes important in carbohydrate digestion include salivary alpha amylase, pancreatic amylase, maltase sucrase, lactase, and alpha dextrinase. Cellulose is a carbohydrate found in plants that provides cell wall structure. This helps maintain plant cell rigidity. Carbohydrates also function to help break down fatty acids and prevent ketosis. Ketosis is a physiological state in which ketone body production exceeds metabolism, resulting in an accumulation of ketone bodies in the blood and tissues. This would result in the use of fat as our main energy source. The ketone bodies produced are then used for a primary energy source. The recommended daily amount of carbohydrates, which is 130 grams per day, would be adequate to prevent ketosis in most people. Ketosis can then have negative health effects, such as kidney stone development, bone/muscle degradation, nutrient deficiencies, and constipation.
In order to understand how our body uses carbohydrates for energy, it is necessary to understand glycolysis, the Krebs cycle, and the electron transport chain. Glycolysis takes place in the cytoplasm, where glucose is converted into pyruvate by a series of phosphorylations. ATP is also produced in this pathway via substrate-level phosphorylation. From there, pyruvate is converted to acetyl-CoA via pyruvate dehydrogenase. Acetyl-CoA is the first precursor to the Krebs cycle. The Krebs cycle is located in the mitochondrial matrix, where we see several electron carriers such as NADH and FADH2 being produced. These carriers will provide the electrons for the electron transport chain. In the electron transport chain, electrons are transferred through a series of 4 complexes. In addition, protons are pumped from the mitochondrial matrix to the inner membrane space. This forms a proton motive force that will fuel the synthesis of ATP through the ATP synthase protein. This form of ATP synthesis is oxidative phosphorylation. When exercising intensely, our preferred energy source is muscle glycogen stores. Blood glucose is typically not used because we want to maintain homeostasis. Eventually, blood glucose can be used during prolonged exercise if we run out of glycogen. When does our body use carbohydrates for fuel during exercise? As duration and intensity increases, our body will utilize more carbohydrates for fuel. Usage of fat as fuel will decrease. Fat is the primary energy source used during lower intensity exercise. Our body's ability to use carbohydrates as an energy source during high intensity exercise is vital. By freeing glucose from glycogen stores, the process of glycogenolysis, glucose can use the anaerobic pathway of glycolysis to produce the ATP. During intense exercise, our body does not have the oxygen present to generate energy through the aerobic pathways, which is why we have adapted to produce those ATP via anaerobic glycolysis. As you can see in this image, lactate will be produced to buffer the acidity caused by the release of protons during glycolysis.
The regulation of blood glucose is important to ensure normal body function. The body depends on a tight control of blood glucose levels. The main stressors for regulation is eating carbohydrates and fasting. When we eat carbohydrates, this stimulates release of insulin from beta cells, allowing cells to take in glucose. This stimulates muscles and liver to store glucose to glycogen and stimulates the storage of amino acids and fat. GLUT4 is a major insulin-sensitive glucose transporter that is found in adipocytes, cardiac muscle, and skeletal muscle cells. When insulin binds to its receptors, there is a series of phosphorylation steps that lead to the translocation of GLUT4 to the cell membrane. This placement of GLUT4 allows glucose to enter the cells and be stored. This transporter has a high Km, which means when the glucose levels are high, its activity is high. When we fast, glucagon is produced by alpha cells, which stimulates the mobilization of amino acids and fats. This stimulates gluconeogenesis and stimulates the liver to release stored glucose through glycogenolysis. There are many other GLUT proteins that aid in the absorption of carbohydrates. Some to review briefly are GLUT1, which provides cells with their basal glucose requirements, GLUT2, which releases glucose from the enterocytes through hepatic portal vein, and GLUT5, which is necessary for fructose transport. Lastly, sodium glucose transporter 1 is necessary for glucose and galactose transport into the enterocyte via the apical membrane. By moving 2 sodium into the cell, glucose or galactose can enter. This transport is active because ATP will be required to return to the sodium-potassium ratio back to its homeostatic state. The acceptable macronutrient distribution range for carbohydrate intake is 45 to 65% of total energy. This range allows for flexibility in the levels because of people's different lifestyles and metabolization of carbs. For those older than 1 year, the dietary reference intake is 130 grams per day, and this is 520 calories of energy. This intake of carbohydrates will prevent ketosis. And lastly, carbohydrate provides about 4 kcals per gram.
Review of ketone body production and effects. This information will become more pertinent later on, but it will be important to understand when ketogenesis occurs and the impact it has on your health. This image to the right reveals that 2 ketone bodies produced are acetone and beta-hydroxybutyrate. These are produced from a precursor of acetyl-CoA, which comes from the breakdown of free fatty acids. Our body enters ketogenesis when there is not enough carbohydrate stores to break down for energy. As a result, free fatty acids are used as energy in the body. This is a result of a low carbohydrate consumption. Recently, there has been a rise in research to determine if ketone bodies are beneficial to individuals. Dr. Pierce will discuss this with us in a future lecture. The short-term side effects of ketosis include fatigue, dizziness, vomiting, low exercise tolerance, headache, nausea, and constipation. These symptoms are usually called the keto flu. The long-term effects of ketosis are hepatic steatosis, hypoproteinemia deficiency, bone degradation, kidney stones, vitamin deficiency, and muscle damage. Now I want to discuss some details on ketosis effects. Kidney stones are at a risk of developing when one is in ketosis due to the decreased pH, or in other words, increased acidity, that the blood and urine will experience. As we know, the function of the kidneys is to help filter acids or alkaline compounds out of the blood by releasing them from the body as urine. During ketosis, the kidneys are filtering the acidic components from the blood, causing a very acidic environment in the kidneys. This leads to uric acid and calcium oxalate crystals. The acidic environment presented with ketosis leads to decreased osteoblast activity. As we know, osteoblasts are the cells that are going to be building bone. In addition, there will be increased osteoclast activity, or bone resorption. This is the cells that break down bone and release calcium. This leads to bone loss, which is a negative effect. In addition, the ketose state can lead to muscle atrophy. One study found that with a low carbohydrate diet, individuals experience reduced muscle mass and reduced grip strength due to the following genes: MAFBX, MERF1, FOXO3, LC3B, and KLF15, which are all going to be upregulated. These genes are specific to muscle atrophy.
This primary research article was conducted by Vitor Virabey, Soledad Arbelazaga, and many others. This research aimed to measure the effect of high carbohydrate intake during ultra-endurance events. The main purpose of the study was to determine if an adequate carbohydrate intake during exercise optimizes athletic performance, limits exercise-induced muscle damage, and improves recovery. This research study aims to compare the effects of high carbohydrate intake and regular carbohydrate intake during ultra-endurance activity, specifically mountain marathons, on exercise load and exercise-induced muscle damage markers. An ultra-endurance event represents one that exceeds more than 6 hours in duration and is one of the major physical challenges for athletes. Exercise-induced muscle damage and internal exercise load are both increased after competing in these types of events. Internal exercise load and exercise-induced muscle damage are the main points researched in this study. Internal exercise load is the relative stressors imposed on an athlete's body during training and competitive events. It has been shown to increase after hard endurance training and competition, representing an increase in internal load and decreased performance. This can be affected by several factors such as rate of perceived exertion, glycogen depletion, heart rate, dehydration, and exercise-induced muscle damage. High-intensity efforts and eccentric contraction induce exercise-induced muscle damage. This can then cause deterioration of muscle function, delayed onset muscle soreness, and increased muscle metabolism, proteins in the bloodstream. During this research study, they collected data on exercise-induced muscle damage markers. These markers include creatine kinase, lactate dehydrogenase, glutamic oxaloacetic, transaminase, urea, and creatine. Higher levels of glycogen ensure adequate muscle function and lower metabolic stress, so it is hypothesized that maintaining adequate muscle glycogen during exercise could limit this exercise-induced muscle damage and improve recovery. The regular carbohydrate recommendation during ultra-endurance events is 60 grams per hour. In order to improve athletic performance in these events, it is recommended to consume 90 grams per hour. With the initiation of nutritional gut training, athletes should be able to tolerate higher amounts of carbohydrates. Nutritional gut training can improve gastric emptying and absorption and likely reduce the chances and/or severity of GI distress, thereby improving endurance performances as well as providing better experiences for the athlete. For example, training an athlete's gut to tolerate high carbohydrate diet will increase the density of sodium-dependent glucose 1 transporters, allowing greater carbohydrate absorption and oxidation during exercise. This has left us to believe that athletes may be able to consume more than 90 grams per hour of carbohydrates during an endurance sport in order to benefit them in the long run. This study was planned as a randomized trial. Carbohydrate intake was compared to an international recommendation of 90 grams per hour of carbohydrates for athletes who exercise for over 3 hours, and regular athletes for carbohydrate intake for 60 grams per hour during ultra-endurance races. In order to participate in the study, you had to be an elite male athlete with at least 5 years of ultra-trail experience and have to undergo nutritional gut training. Gut training consists of ingesting 90 grams per hour of carbohydrates at least 2 days per week in the 4 weeks prior to the mountain marathon. All runners were examined prior to the study for disease or injury that could alter the research results. These 20 male athletes were then split into 3 groups: 7 athletes in the experimental group, which consisted of the athletes ingesting 120 grams per hour of carbohydrates, 7 athletes in the control group, which consisted of the athletes ingesting 90 grams per hour of carbohydrates, and 6 athletes in the low group, which consisted of the athletes ingesting 60 grams per hour of carbohydrates. These 3 groups took the same carbohydrate form during the mountain marathon. This carbohydrate concoction was 30 grams maltodextrin, also known as glucose, and fructose gels, a ratio of 2:1. The ratio of glucose to fructose was specifically made for this study because it was used to increase exogenous carbohydrate oxidation during exercise. Glucose was then able to be absorbed by the sodium-dependent SGLT1 transporter, and fructose was absorbed through GLUT5 transporter. An increased amount of carbohydrate intake can limit limit its absorption and subsequent oxidation, so this way there was another path for carbohydrates to be absorbed with little to no GI discomfort. The carbohydrate intake protocol was programmed for every 15, 20, 30 minutes, with these athletes consuming 1/4, 1/3, or 1/2 of the total carbohydrate amount per hour according to the experimental, control, and low groups, respectively. These participants were not able to consume any other food during the mountain marathon, just water as needed. Internal exercise load was then calculated using the individual session rate of the perceived exertion method. This method was determined as being the product of mountain marathon finishing total time times the subsequent rate of perceived exertion. The athlete's overall difficulty in the mountain marathon was recorded at the same time as the race ended. All athletes also had to participate in a blood test at 2 specific points during the research. The first blood extraction was the day of the race, and the second was 24 hours after they had completed the mountain marathon. The blood sample analysis tested for serum markers of exercise-induced muscle damage and protein catabolism markers. Along with the blood test, every athlete had personal diets and menus prepared specifically for them. They started these menus 48 hours before the start of the mountain marathon. This was done in order to optimize their glycogen content with 9 grams of carbohydrate per body mass per day, 1.5 grams per kilogram per day of protein, and about 0.5 grams of fat per kilogram of body mass per day. They could not eat any butter or fatty meat to avoid any interaction between dietary intake and exercise-induced muscle damage. The runners ate a specific breakfast the day of the event that was calculated out for each runner. They then wore GPS alarms based on their study group to notify them when to consume the carbohydrate concoction throughout the race. At the end of the race, they were given a protein shake equivalent to 1.2 grams of carbohydrate per kilogram with 0.3 grams per kilogram body weight of whey isolate protein. The athletes then consumed a high carbohydrate meal within 24 hours of completing the race to replenish glycogen stores in the body. After all of this was done, statistical data was analyzed using SPSS statistics software with statistical significance designated when P was less than 0.05. Here we can see that figure 4 shows that there's a significant difference in exercise load where P equaled 0.019 specifically in the experimental group. Figure 5 then shows that there were significant differences between groups in the percentages change in glutamic oxaloacetate, transaminase change with a p-value of 0.003, lactate dehydrogenate change with p being less than 0.001, and creatine kinase change with p being 0.019. Overall, this study aimed to compare the effects of different carbohydrate intake during a mountain marathon on exercise-induced muscle damage and exercise load. This research study hypothesized that a 120 gram per hour dose during a mountain marathon could limit exercise-induced muscle damage markers and improve muscle recovery in ultra-endurance athletes. This study found that a higher carbohydrate intake compared to the current recommendations during a mountain marathon significantly limits the increase in exercise-induced muscle damage markers. They also saw a significantly lower exercise load was found in the experimental group compared to the low and control groups. This shows that a higher carbohydrate intake could be a determining factor in the internal exercise load response. Additionally, these findings show that the current recommendations of 90 grams per hour of carbohydrates for exercise lasting longer than 2.5 hours might not be enough to limit physiological and metabolic responses after a mountain marathon.
Now that we've discussed carbohydrates as it relates to endurance athletes, we wanted to look at how these carbohydrates translated to strength athletes as well. This article was conducted at the School of Exercise Science by Australian Catholic University. It was published in the Journal of Strength and Conditioning Research in December of 2018. Most studies that explore this topic of low-carb, high-fat diet and exercise have primarily been explored in recreational athletes. The people who conducted this study were looking to see the efficacy of this diet among long-time Olympic level and professional athletes in the world of strength and weightlifting. As I mentioned previously, much of the debate surrounding high and low carbohydrate diets for athletes typically relates to endurance athletes such as cyclists and runners. This is exactly what the research wanted to emphasize in their study. A huge advantage of sports like these is the division of weight classes. Unlike endurance sports, many strength sports are separated into weight classes and judged accordingly. If this diet proves to be successful, it could potentially be used to reduce body weight thus an athlete's weight class. If their strength is found uncompromised, then this could be a useful tool for weightlifters and bodybuilders to use. The researchers hypothesized this by stating that a low-carb ketogenic diet would result in meaningful reductions in body weight with lifting performances compared with the usual diet. In order to be eligible for this study, the researchers wanted any person within the age range of 18 to 55 with at least 6 months of lifting experience. The researchers really specifically wanted not just Olympic and professional weightlifters, but those who have competed at local and national levels and who have been lifting for a little bit over 1 to 13 years. 14 athletes were used in this study, while 16 were assessed, because two were ruled out through the accessibility rules. In this study, 5 were female and the remaining 9 were male. Ages ranged from 24 to 53 years of age. A 12-week diet period was the start of this study, with half of them being in the low-carbohydrate ketogenic diet, while the other half were in the usual diet. Followed by that is a 14-day washout period, and then they would switch the diets to be the opposite for the remainder of the 12 weeks. The two diets being compared, again, are the low-carb ketogenic diet compared to the usual athlete's diet. As for the methods, this was a randomized crossover study with 14 intermediate to elite level Olympic power and weightlifters. The data collected was separated into 2 sections, one to be collected by the university, while the other to be self-reported. By the university, they would be assessing lifting performance, body composition through DEXA scan, resting metabolic rate through indirect calorimetry, blood glucose levels, and blood electrolyte levels. These were collected at baseline, 3 months, and 6 months, and the participants had to be in a fasted state. As well as doing self-reported data, the participants were doing dietary intake through MyFitnessPal, self-monitored blood glucose and blood ketones by doing a finger prick at home, as well as weekly training measures, which they were just keeping in a journal. They were doing this 3 times a week. Once again, the 2 diets followed are a low-carb ketogenic diet, with the target range being 70% fat, 20% protein, and less than 10% of carbohydrates. Usual diets were just instructed to continue their normal diet. However, in this crossover study, the participants who started with the low-carbohydrate diet and then switched to the usual diet after 12 weeks were instructed to ensure they get at least 250 grams of carbohydrates per day to mitigate the carryover effect. What's interesting about this study is that they had to continuously push back the start of the study in order to make sure that all athletes were on the same training schedule. As we know, for sports like weightlifting, is that they go through phases such as cutting, bulking, or maintaining. And some of the athletes for this study were in different phases. So they made negotiations and also altered the start date in order to make sure that all athletes were in the exact same phase. Although there were many key data points collected in this study, I just wanted to cover a few to get the basis of the study. First is the athlete's dietary intake, which we'll discuss shortly, which again was recorded using MyFitnessPal by the subjects. Body composition was found using the DEXA scan provided at the university's lab. And next, they studied the lifting performance, which was a 1-rep max. And I found it interesting that the subject gets to pick the lift in which they are recording that 1-rep max. This was ensured to be able to make sure that the subjects did not do a move that they were unfamiliar with. So the researchers allowed them to pick whatever moves they were comfortable with to ensure that they had a good performance. Lastly, blood ketones were also measured, and again, this was measured at home using a portable analyzer with a finger prick. Again, there were many key aspects studied, but we just wanted to cover a few main ones. Firstly, we wanted to look at the 2 diets specifically with remaining the same. Total energy intake and protein intake relatively remained the same across the usual diet and the low-carb ketogenic diet. The significant differences, however, which we can imagine is the carbohydrate and fat intake, as you can see from the very top column, where the athletes were consuming over 200 grams of carbohydrate in their usual diet to about 40 grams in the low-carb diet. Same with fat. We can see that they were consuming around 80 grams of fat in the usual diet and around 163 grams in the low-carb ketogenic diet. Next, we want to look at the changes in body mass, specifically in the overall body mass as well as lean body mass. The change was significant in the overall body mass with an average of 3.26 kilograms lost. This includes an average lean body mass of 2.26 kilograms as well, which we all know is undesirable, especially for strength athletes, while fat mass stayed relatively the same. This creates more of a discussion on how the body utilizes fats and proteins when glycogen stores deplete. So why did lean body mass decrease while fat mass remained unchanged? Lean body muscle loss comes from the release of glutamine and alanine from the skeletal muscle during prolonged fasting. These are then used in the gluconeogenesis pathway. Although protein only made up about 20% of the diets, it is worth noting this since the majority of the diet was derived from fats. As for training remained relatively unchanged in sessions, minutes per week, and training load. This means that there is an ability to decrease their body mass and possibly even a weight class while also keeping their desired strength levels. This article furthers the possibility of these diets when weightlifters are in certain cycles of their training. Lastly, also not shown on this graph, is blood ketones, which were slightly elevated from the baseline, which would be expected considering the diet was, again, derived from 70% of fats. And thanks to Mallory covering this on the previous slides, we know that two ketone bodies are produced, acetone and B-hydroxybutyrate, which are derived from the breakdown of free fatty acids to acetyl-CoA, better known as the beta oxidation cycle. It's also worth noting overall, however, that this was done on a small group of only 14 people and only within a span of a few months. Prolonged carbohydrate depletion would create more of an emphasis on gluconeogenesis in the body. It would be interesting to see further research in this field, specifically as to how it favors protein and or fat stores once glycogen has depleted.
This research article was conducted by Nadine B. Washmuth and Felix Aber, along with many others. The purpose of this study was to investigate the impact of performance and body composition in physically active adults on a high carbohydrate/low fat versus low carbohydrate diet. We need to start by looking at what a low and high carbohydrate diet are and the initial effects they have on the body. A low carbohydrate diet is one where the carb intake is less than 10% and 26% of the total caloric intake. 10% being 20 to 50 grams and 26% being less than 130 grams. Also, with a low caloric intake of carbs in a low carb diet, the energy source should be shifted to protein and fat. The body has to find alternative ways to produce energy when there is not an abundance of carbs to use. When intake of carbs is low, ketosis occurs, causing glycogen to plummet so hypoglycemia doesn't happen. Ketones are made when the body has to turn to fat to use as fuel, and then the ketones distribute energy. After 48 hours of following a low-carb diet, gluconeogenesis occurs in the body to make sure glucose is being given to the central nervous system and red blood cells. For energy to continue to be made, the blood takes fatty acids to the muscle and liver. Some research has shown that during a low-intensity prolonged exercise sessions, a low-carb diet can delay exhaustion as well as cause fat loss without hurting performance. It has also been shown that low-carb diets help those with obesity during aerobic exercise by encouraging fat oxidation in the body and lowering body fat. A high-carb diet is one where the carb intake is higher than 45% of total energy intake. It is the general recommendation to consume carbs as a way to help performance because carbs are the only macronutrient that are absorbed right away to be used for energy. Performance was evaluated during pulmonary exercise testing and studied peak oxygen uptake and peak performance. Also, body composition was investigated as an outcome of performance being studied. Materials and methods. This study was a single-center, non-randomized, crossover, controlled pilot trial. There were 24 physically active adults between the ages 18 to 41 years old, with 14 being female and 10 being male. 18 participants in the intervention group were following the low-carb diet and high-carb diets, and 6 participants were a part of the control group. The participants had normal nutritional behavior and a body mass index between 18 and 27. The first 3 weeks of the study, the intervention group followed a high-carb diet defined as 75 to 80% carbs, 15% proteins, and 5 to 10% fat. Complex carbs were the main source of carbohydrates in the diet. The participants avoided carbs containing sucrose and fructose. After the 3 weeks of following the high-carb diet, there was a 3-week washout period where participants returned to their normal diet. Then, once the 3-week washout period was over, the intervention group began to follow a low-carb diet. The first week was a low-carb diet being 20 to 25% carbs, 15% protein, and 60 to 65% fat. Then, in the last 2 weeks, the group followed a ketogenic diet being 5 to 7% carbs, 15% proteins, and 80% fat. The control group was studied during the same time period, but did not have to follow any specific nutritional advice. Aerobic performance, resting metabolic rate, body composition, daily activity and nutrition, and venous blood samples were all collected throughout the study. Methods. After each 3-week period of the high-carb and low-carb diets, aerobic performance was tested with the cardiopulmonary exercise test on a cycle ergometer. There was a 3-minute warm-up phase at 50 watts, and the load would increase 17 watts every minute. The load continued to increase until the participant decided that they had reached exhaustion. Respiration was measured breath by breath, and the capillary blood samples from the earlobe were taken to measure lactic acid and blood glucose concentrations. VO2 peak, performance peak, and oxygen efficiency were all measured. Resting metabolic rate was also measured after each 3-week period of the high and low carb diets. RMR was taken with the participant resting for 30 minutes in supine position. This rate measured the total number of calories burned when the body is at rest. Body composition was taken weekly during each trial. Bioelectrical impedance analysis was used to measure body mass, fat mass, skeletal muscle mass, and total body weight. Daily activity and nutrition were collected for 1 week in each 3-week trial. The daily physical activity was measured through a continuous Bluetooth activity monitor that the participant wore at all times throughout the day. Macronutrients were analyzed from food diaries filled out by the participants. Lastly, the venous blood samples were taken at the end of each 3-week trial. Ketone levels, C-reactive protein, interleukin-6, and lipids, including total cholesterol, HDL, LDL levels, and triglycerides, were all analyzed. During the cardiopulmonary test, there was no significant differences in peak oxygen uptake and heart rate when comparing all 3 groups. Some significant changes that did occur were the peak performance was found to be much higher for those following a high-carb diet. In the control group, peak performance was still much higher than both diet trials. And when measuring oxygen efficiency, it was higher after a low-carb diet than after a high-carb diet. Time to exhaustion was longer after the high-carb diet compared to the low-carb one. It was much longer for the control group when being compared to both diet trials. Also measured for performance parameters were blood lactate levels and blood glucose levels. Blood lactate levels were not significantly different between the 2 diet trials, but when compared to the control group, levels were higher after a high-carb diet. Blood glucose levels were higher in the high-carb diet compared to the low-carb diet. There was no significant change in lean body mass, skeletal muscle mass, resting metabolic rate, and physical activity, or ingested activity calories when comparing all 3 groups to each other. When looking at body mass, both diet trials resulted in major decrease of body mass, body fat percentage, and visceral fat from the baseline to after 3 weeks. As for nutrition, caloric intake was lower in the high-carb diet trial compared to the low-carb diet trial. Carbohydrate intake was, of course, higher in the high-carb diet, but lower for fat and protein intake compared to the low-carb diet. Next, we can take a look at the biochemical parameters. There were no significant changes for the control group, but there were for the 2 diet trials. The only big changes in the low-carb trial was that there was an increase in ketone levels. In the high-carb trial, there was a decrease in cholesterol levels, low-density lipoprotein levels and high-density lipoprotein levels. And there were no significant changes in C-reactive protein and interleukin-6 levels for any of the groups. The purpose of this study was to look at the impacts of high versus low carbohydrate diets on physical performance and body composition. Through the 3-week trials of a high-carb diet and a low-carb diet, physical performance was impacted positively by the high-carb diet. This was shown from the results of higher time to exhaustion and higher performance peak. Body composition was changed at the end of both diet interventions pretty similarly. In the high-carb diet trial, it showed a major decrease in total and LDL cholesterol, while there's an increase in triglycerides. In conclusion, although there were many results that did not have enough of a significant change to show much impact on the participants' performance or body composition, there were still a few positive changes that resulted from a high carbohydrate diet.
Now, it is important to consider other health factors. Athletes typically want their diet to support their overall performance, which we have seen in the past 3 studies that we talked about. But what about the effects that low and high carbohydrate diets can have on other aspects of their health? In the final research article that will be presented, I'm going to discuss the impacts on your kidneys, bone, and muscle. This final research article is titled Chronic Ketogenic Low Carbohydrate High Fat Diet, which has minimal effects on acid-base status in elite athletes. This primary research article was conducted by Amelia Carr, Avish Sharma, Megan Ross, along with many others. The goal of this research was to determine the effects of a sustained low carbohydrate diet on elite athletes' acid-base balance. In addition, it was determined, can the net endogenous acid production explain these changes in acid-base balance? Here is some background information that will be helpful with understanding this study. So to better understand this study, it is necessary to know why we want to maintain an acid-base balance. When disrupting our body's acid-base balance, we can experience unnecessary stress factors that can lead to kidney stones, bone damage, muscle damage, and more. This is why we reviewed briefly the process of ketogenesis previously and the effects it has on our health in this presentation. Our body can experience this acid-base imbalance from many different origins, but one specifically is going to be our nutrition or dietary intake. It is important to understand how dietary intake can affect the blood's pH. When consuming a high-fat diet, one's pH will decrease, or in other words, become more acidic. In addition, a low-carbohydrate diet is associated with a decrease in blood pH when compared to the blood pH of a high-carbohydrate diet. The research that has been conducted on these effects thus far was from only acute dietary changes and did not investigate the effects in athletes, which is why this research study took the time to determine the effect of chronic dietary alterations on acid-base balance in athletes. The methods of this study. So in this study, it was a non-randomized parallel group study design, which was used to evaluate the effect of 3 different sustained diets on elite athlete race walkers and their acid-base balance. The effects were followed at rest and after a graded exercise test. In this study, there were a total of 24 participants, 17 males and 7 females, that were all elite level race walkers. The 3 differing dietary interventions were followed for 21 days each. 9 participants were tasked to follow a low carbohydrate diet and high fat. This entailed a consumption of less than 50 grams carbs, 15 to 20% of protein, and 75 to 80% fat. 7 participants followed a periodized carbohydrate diet. This periodized carbohydrate diet focused on having high carbohydrate diet prior to endurance training while consuming a lower carbohydrate diet when athletes are partaking in lower intensity sessions. In the previous research, this method has revealed performance benefits in those who are sub-elite athletes. So individuals that were partaking in the periodized carbohydrate diet were sub-elite athletes that had shown benefits. Lastly, 8 participants were used as the control, where they followed a high carbohydrate diet. So the macronutrient breakdown was 60 to 65% of energy intake from carbohydrates, 15 to 20% from protein, and 20% from fat. The assignment of these diets was based on the participants' most desired diet to follow, so that made this a non-randomized trial. There were many data points that were collected. So the data collected in the study was blood pH, blood bicarbonate, and blood lactate. Each were collected via capillary blood samples. As stated previously, these measurements were taken pre and post an exercise test. We'll see this later on, but the main data that we're looking at is going to be blood pH. Additional data was collected prior to the completion of the exercise test. Participants' net endogenous acid production, or NEAP, was determined from 48 hours prior to the start of the dietary intervention. In addition, the NEAP was determined from 72 hours of dietary intake. These 72 hours were the final 3 days of their participation in the dietary interventions. For both data collection times, mean values were utilized to account for any variation that had occurred due to their dietary intake. The data was collected at 2 different pinpoints in time. To start, the participants' data was collected. This means no dietary intervention was implemented and the participants followed a dietary pattern of their choice. Then the data was collected once the participant followed the intervention and all food following the dietary pattern was consumed under supervision of a dietitian. This allowed accuracy. The capillary blood samples were taken prior to the exercise test, and then they were also taken post 2, 4, and 6 minutes after the maximal exercise test. All blood tests and NEAP data collected were then analyzed using a general linear mixed model. As with all research studies, it is important to control any and all confounding variables. In order to control the dietary intake of participants, all meals were prepared by a chef that allowed standardized recipes that followed the dietary interventions of each participant. Each meal was consumed in a group environment, so intake could be monitored by a registered sports dietitian. In addition, participants were able to request additional food when they were showing signs of hunger. The food that was provided additionally would still follow the macronutrient distribution that followed the specific dietary intervention of the participant. All food consumed by participants would then be recorded and accounted for in a log. Energy and nutrient intake would then be calculated from a food analysis database by the registered dietitian that supervised the mealtimes. Apart from dietary intake being controlled, the training intensity and quantity had to be controlled. Participants participated in 12 weekly training sessions, 6 of which were race walking that was mandatory and supervised within a group. These supervised sessions varied in intensity, duration, elevation, and more. The other 6 sessions could be chosen by the participant. They could partake in more race walking, cross-training, or whatever other option they chose from, but it was required that they recorded these individual trainings on a log. Now, the controlling of the graded maximal energy test that each participant went through was performed on a motorized treadmill that was custom built, made specific for this study. The test had 4 different stages that were submaximal. These were used to determine the participant's submaximal VO2 and walking economy. Each stage lasted 3 minutes long and then was followed by an incremental ramp to determine the participant's VO2 peak. With each stage being completed, it took participants roughly 13 to 20 minutes. Now it is important to discuss the results of this study and the information that is pertinent to why we chose this research. So pH is the data collected that we are most focused on for this research study. It was shown that at a baseline, there was no significant differences in blood pH between the 3 dietary interventions. After the exercise test, there was a significant decrease in pH for all dietary intervention groups. And after all the interventions were completed, there was no significant difference in the blood pH between the groups. For both bicarbonate and lactate, there was no significant difference in blood bicarbonate, blood lactate levels for both pre-exercise or post-exercise between groups at baseline and the post-training intervention. For the post-intervention net endogenous acid production, the NEAP, the low carbohydrate, high fat diet, the NEAP values were unchanged. Significant decreases were seen in the NEAP values for the high carbohydrate diet and the periodized carbohydrate intervention groups. But there was no significant difference between these two groups. The NEAP was higher in the low carb, high fat compared to the control, which, as we know, the control was the high carbohydrate diet. Now I want to review the important findings and overall conclusion of this study. So when participants followed a low carbohydrate, high fat diet, there was no significant difference in the blood pH, bicarbonate, and lactate levels when compared to the participants that followed the high carbohydrate diet. Now, as previously stated, it was seen that those who followed the low carbohydrate, high fat diet had a higher post-intervention NEAP. This trend was not seen in the post-intervention for either blood pH or bicarbonate levels, despite knowing that there was ketone body production that would increase with following this diet. This may be explained indirectly by the individual's well-developed blood buffering capacity. In other words, with more research, we could conclude that the NEAP may not be relevant for elite athletes. The overarching result. When following a low carbohydrate dietary intervention, elite athletes will not experience any additional impacts on their acid-base status compared to those who followed a more carbohydrate-rich diet. Explanation. Athletes likely have experienced extreme conditions in previous training, which has led to adaptations in their body's ability to manage blood pH levels. In other words, they have greater buffering capacity, respiratory, and renal pathways. These adaptations may explain why there were no changes seen in the post-intervention acid-base status among the differing dietary interventions. As with all research, it is important to understand the limitations. This study had a limited sample size due to limited resources. They also had a definition of an elite athlete that could not qualify some individuals to partake in the study, and more. Additional research would be beneficial to determine the effects of dietary interventions on net endogenous acid production and acid-base balance. Considerations. Although the study revealed that a low carbohydrate diet will not likely impact an athlete's bone, muscle, or kidneys, it is important to understand that this was only in relation to the dietary intervention's impact on blood pH. A low carbohydrate diet can lead to a decrease in calcium consumption from one of the main contributors, dairy products. As we know, dairy does contain carbohydrates, so with a low carbohydrate diet, this will likely be eliminated. A low carbohydrate diet can also lead to decreased intake of magnesium-rich whole grains, which will impact bone health as well. Magnesium helps build the bones and the crystal lattice and the bone surface. So with a decrease in intake, bone can experience a loss of integrity. Muscle health can also be impacted by a decreased carbohydrate diet. Vitamin C plays a role in collagen synthesis by activating the enzyme hydroxylase, which helps the cross-linking occur for stability. Vitamin C-rich foods include many berries, citrus fruits, and vegetables. These are higher in carbohydrates than other dietary options, which means they are likely limited in a low carbohydrate diet. Without adequate vitamin C intake, muscles may experience deterioration. A low carbohydrate diet can also impact kidney health. Magnesium, potassium, and cobalamin all may decrease with a low carbohydrate diet. Each of these micronutrients play a role in protecting the kidneys against chronic kidney disease. It is necessary to do further research to assess other impacts of low versus high carbohydrate diets on the micronutrient intake because many micronutrients play a key role in bone, muscle, and kidney health. Overall conclusions from all 4 of our primary research studies. High and low carbohydrate diets have positive impacts on some aspects of health for athletes and negative impacts on others. It is very important to consider the athlete's overall goal before deciding on the composition of their diet. For example, if an athlete is wanting to increase performance rates, following a high carbohydrate diet has been shown to improve an athlete's time to exhaustion in one of the research studies that we had presented. On the other hand, if an athlete is considering a low carbohydrate diet, we do know an athlete will not have additional impacts on their muscle and bone health compared to someone who were to follow a high carbohydrate diet. Basically, they're at an even playing field, which is beneficial to know that their performance will not be impacted anymore in considering muscle and bone health compared to those who follow a high carbohydrate diet. Lastly, we have some discussion questions to share with our peers to answer in our discussion board. 1. What do you think would happen if GLUT5 is not able to aid in the absorption of carbohydrates? Do you think that this would change the effective carbohydrate intake for exercise? Explain. 2. How would you define a low carbohydrate diet versus a high carbohydrate diet? What guidelines would you set? Why do you think lower carbohydrate diets would work well for weightlifters versus endurance runners? Do you think a normal diet would still work to see adequate strength? 4. And last one, can you think of any other nutrient deficiencies or complications that may be present with a low carbohydrate diet that would impact health in any manner? Explain. And here are all of our references for this presentation. Thank you for listening, and I hope you guys learned something new today.